Hello students, I am Dr. Amit Bhatnagar, working as Senior Research Officer in the Department of Agronomy at GB Pant University of Agriculture and Technology, Pantnagar, Uttarakhand. What is DPD? Diffusion Pressure Deficit. It is very important for the calculation of water potential. Actually, water potential is very confusing and we have confusion in osmotic potential, turgor pressure, metric potential, gravitational potential and total water potential. And this DPD is very important parameter which decide the water potential. So what is the concept of this DPD? If we want to know the actual water potential of the cell, we must know what is diffusion pressure deficit. Actually, this diffusion pressure deficit decide the water potential, and we know that water potential of any cell is equal to negative of this DPD. So negative value of diffusion pressure deficit is the water potential of cell, and the symbol of water potential is psi. This is psi, PSI. This psi symbol is PSI and it is denoted as PSI W. So water potential is equal to minus DPD. So we should know what is DPD. Without knowing DPD, we cannot know about the water potential. तो ये डिफ्यूजन प्रेशर डिफिसिट क्या है? एक्चुअली वाटर पोटेंशियल जो होता है वही डिफ्यूजन प्रेशर डिफिसिट होता है, जो उसकी नेगेटिव वैल्यू होती है. तो हमको पहले जानना पड़ेगा कि ये डीपीडी क्या है. बिना इसके जाने हम वाटर पोटेंशियल नहीं जान सकते कि किसी सेल का वाटर पोटेंशियल क्या होता है. तो ये क्या है डीपीडी, इसको जानना हमें जरूरी है. तो नाउ इट इज क्लियर दैट डीपीडी इज द वाटर पोटेंशियल ऑफ cell. So how this DPD is calculated and what is the meaning of this DPD? Actually, this consists of two terms, diffusion pressure and deficit. Deficit means reduction. So reduction or decline is called the deficit. And the diffusion pressure, and diffusion is the movement of water from high water potential to low water potential or less concentration to high concentrated solution. So please watch the lecture Process of Osmosis already uploaded in the playlist General Agriculture. So please watch the Process of Osmosis and another lecture Osmotic Potential. So suppose this is the water and this is the pure water in the cell. Water is present in this and it is separated by pure water is separated by another cell, and this is a semi-permeable membrane. In between there is a semi-permeable membrane. This is semi-permeable membrane and water is also present here, and this is also pure water. Suppose then water is moving across both sides, left to right and right to left. But we add some salt. We add some salt we are adding some salt or sugar or electrolyte which is soluble. Then here the water start moving from pure water to this solution. So now it is solution because it has water plus salt. Water plus salt or solute, we can say solute. Solute may be salt or electrolyte or sugar, and here it is pure water. So now the water is going or moving from pure water to this solution. अगर दो सेल सेपरेटेड हैं सेमी परमेबल मेम्ब्रेन से, तो प्योर वाटर कहाँ मूव करता है? सॉल्यूशन की तरफ मूव करता है. ये आप जो प्रोसेस ऑफ ऑस्मोसिस है उसको देखेंगे, डिफ्यूजन को देखेंगे तो पूरा आपको समझ में प्रोसेस आएगा. उस लेक्चर को आप जरूर देखिए. अब क्या है कि ये एक प्रेशर क्रिएट कर रहा है. नाउ दिस water is moving with the pressure into this cell. Now, so there is a diffusion pressure by which water is moving from pure water to this solution. Now, suppose we add some salt in this pure water. Now the diffusion pressure is reduced. Now water will move from this solution to this solution. This is also a solution now. This is also solution but less concentration, and it is high concentration. Here we are adding only small amount of salt, here more amount of salt. Now तो वाटर विल मूव फ्रॉम दिस लेस कंसंट्रेटेड सॉल्यूशन टू हाई कंसंट्रेटेड सॉल्यूशन, बट द प्रेशर बाय विच इट इज मूविंग और डिफ्यूजिंग इज रिड्यूस्ड। अगर इस प्योर वाटर में कुछ साल्ट मिला दें तो अब ये वाटर तो मूव करेगा, लेकिन उसका जो मूव करने का जो प्रेशर, जो मूव कर रहा है, जो डिफ्यूज हो रहा है, वो प्रेशर रिड्यूस हो गया क्योंकि हम साल्ट इसमें ऐड कर रहे हैं। और जैसे जैसे हम साल्ट ऐड करते हैं ये डिफ्यूजन प्रेशर रिड्यूस होता जाता है। एज वी इंक्रीज द अमाउंट ऑफ साल्ट द प्योर वाटर and its pressure decreases, diffusion pressure decreases, and this is called diffusion pressure deficit. It means when we add some salt in pure water, its diffusion pressure decreases and it is directly proportional to the amount of solute or salt. तो जैसे जैसे हम वाटर के अंदर साल्ट मिलाते जाएंगे उसका डिफ्यूजन करने का जो प्रेशर है या जो एबिलिटी है वो क्या होती जाएगी, कम होती जाएगी, एंड दिस इज नोन एज डिफ्यूजन प्रेशर डिफिसिट। ये अब आपको समझ में आ गया होगा कि व्हाट इज डिफ्यूजन प्रेशर डेफिसिट। सो दिस इज द डिफरेंस, वी कैन से दिस इज द डिफरेंस बिटवीन द डिफ्यूजन प्रेशर ऑफ अ प्योर वाटर एंड द सॉल्यूशन, डिफ्यूजन प्रेशर ऑफ सॉल्यूशन। फॉर एग्जांपल मैं आपको दूसरा एग्जांपल भी एक बता देता हूँ कि सपोज देयर आर टू cells are there and pure water is present, pure water, and here also pure water. Okay, and they are in a contact with the cell with the semi-permeable membrane. This is the semi-permeable membrane difference. This is the semi-permeable membrane, and here solution is there. Suppose a 2% solution, 1 mole solution, 1 mole, suppose for 1 mole solution is there of NaCl, suppose for example. Here also 1 mole NaCl solution, and the amount is same. Amount is same. So in both the case, pure water is present and 1 mole solution is there. So diffusion is start from pure water to this 1 mole solution. This is the process of diffusion. But now diffusion pressure is equal. So this is the diffusion pressure of pure water in both the case A and B. Both the case, suppose this is the A cell and this is the B cell. In both the case diffusion pressure is same. All the things are same. Now we are adding some salt. Suppose in this we are adding some salt. Suppose you are adding 0.2 mole NaCl. Now the diffusion pressure will decrease as compared to this A cell because some osmotic pressure is also maintained. So we are adding some salt, so the capacity of free movement of water is reduced. So there is a reduction in diffusion pressure because some salts are present which restrict the free movement of water. तो अगर हम दोनों प्योर कंडीशन सेम है और इसमें हम कुछ साल्ट मिला दें तो इसके कंपेरिजन में इसका डिफ्यूजन प्रेशर कम हो जाएगा क्योंकि इसमें साल्ट और साल्ट वाटर के साथ इंटरेक्शन शो करेंगे, उसके फ्री मूवमेंट को कम कर देंगे। तो यहाँ डिफ्यूजन प्रेशर कम हो गया एंड दिस इज नोन एज diffusion pressure deficit. So this DPD is the difference between diffusion pressure of pure water and diffusion pressure of this solution. So as we increase the amount of salt, suppose now this is 0.5 molar, then further reduction in diffusion pressure. Suppose it is 0.8 molar, then further reduction in diffusion pressure. So as we increase the amount of salt or solute, the diffusion pressure deficit increases, or we can say there is more and more reduction with adding amount of the solute. तो जैसे जैसे हम solute मिलाते जाएंगे डिफ्यूजन प्रेशर घटता जाएगा। ये आपको एक एग्जांपल मैं समझता हूँ आपको अच्छी तरह से समझ में आ गया होगा। So this DPD is in with reference to A and B. This is the diffusion pressure of pure water. This is the diffusion pressure of the solution. So this is known as DPD. So this DPD is very important in deciding water potential. Actually this DPD decide the intake of water. Here there is more intake because more diffusion. Here there is less intake because less diffusion of water. So this is the DPD. तो अब आपको समझ में आ गया होगा कि डिफ्यूजन प्रेशर डेफिसिट क्या है। जैसे जैसे हम प्योर वाटर में साल्ट मिलाते जाएंगे उसका जो डिफ्यूजन का प्रेशर है वो क्या होता जाएगा, कम होता जाएगा, सो मूवमेंट ऑफ वाटर, फ्री मूवमेंट ऑफ वाटर reduces and its capacity to diffuse reduces. So that's why its capacity to enter into the other cell decreases. So it is also known as suction pressure. This is the suction pressure. Suction means to suck, किसी चीज को खींचना, वो that is called suction. So to suck, the capacity to suck the water is reduces, so it is known as suction pressure. And this dpd is calculated as osmotic pressure minus turgor pressure. Now what is osmotic pressure or turgor pressure? So please watch the lecture, what is osmotic potential or osmotic pressure. Briefly मैं यहाँ फिर से इसको बता दे रहा हूँ. Suppose this is a container, this is in U form and water is present in this U tube, we can say tube. Water is present, freely moving, and there is a semi-permeable membrane, so there is free movement of water both sides, and we add some salt, definitely water will move from this direction to this direction. So the pressure which is created during the osmosis, during the diffusion, is called the osmotic pressure. Okay, so this is the osmotic pressure which is created, which is developed during the osmosis. Or in other terms, suppose there is a piston, and we want to check this diffusion. So if we create a pressure so that entry of water is checked, so that pressure which is required to check the entry of water is known as osmotic pressure. तो osmotic pressure वो pressure है जिस pressure से water जो है pure water किसी solution में move करता है through semi permeable membrane. या हम ये कह सकते हैं कि इस water की entry को रोकने के लिए हमें ऊपर से जितना pressure लगाना पड़ेगा, that is osmotic pressure. So this is the osmotic pressure. Now what is the turgor pressure? Turgor is related with the turgidity. Suppose it is a cell, and in cell cytoplasm is present and water is also present, and this water in cytoplasm exert a pressure on the cell wall in normal condition. So this pressure of water of cytoplasm which is exerted on cell wall is known as turgor pressure. So turgor pressure is the pressure which is developed by the water or which is exerted by the water on the cell wall is known as turgor pressure. As the pressure increases, cell swell more and more. So as turgor pressure increases, cell size increases. So cell size increases due to turgidity and due to turgor pressure. So this dpd is the difference of osmotic pressure minus turgor pressure. So turgor pressure क्या प्रेशर होता है कि सेल के अंदर जो पानी है वो सेल वॉल पे कुछ प्रेशर लगाएगा नॉर्मल कंडीशन में भी, तो उस प्रेशर को हम टर्गर प्रेशर कहते हैं. और जैसे जैसे सेल फूलती जाएगी, बड़ी होती जाएगी, उसका वाल पे प्रेशर क्या होता है, बढ़ता जाएगा. और जो ये वाल है वो अपोजिट डायरेक्शन में प्रेशर लगाती है, इक्वल एंड अपोजिट, दैट इज नोन एज वाल प्रेशर. तो यहाँ जो टर्गर प्रेशर होता है वाल प्रेशर के इक्वल होता है. सो व्हेन वाटर एक्सर्ट प्रेशर ऑन द सेल वॉल, इट इज नोन एज टर्गर प्रेशर, and in turn cell wall exert the pressure opposite and equal amount, that is known as wall pressure, because it is exerted by cell wall, that's why it is known as wall pressure, and it is due to turgidity, that's why it is known as turgor pressure. Okay, so this is the formula of this dpd. Suppose a cell is there and it is exerting pressure on the cell wall, water is present, and this is known as Tp. And now cell is fully turgid, means it has maximum capacity. Fully turgid cell. So maximum water content is there and it exerts pressure on cell wall. So in this case, if water movement is from outside, there will be no entry of water due to osmotic pressure is suppose there. So since maximum pressure is there, it has maximum intake of water. So the osmotic pressure, suppose a water cell is in another solution, so osmotic pressure is by another cell, but it has maximum water potential, maximum water content, and there will be no entry because there is a fully turgid. अब इतनी fully turgid cell है कि अंदर का प्रेशर बहुत ज़्यादा हो गया है, मैक्सिमम स्टेज पे है, तो ऑस्मोटिक प्रेशर से जो पानी इसके अंदर आ रहा है वो नहीं आएगा. उस केस में क्या होगा, OP इज इक्वल टू osmotic pressure is equal to turgor pressure. So in this case, in fully turgid cell, dPd, since OP minus TP and OP and TP are equal, so dP, diffusion pressure deficit, is equal to 0. So in fully turgid cell, diffusion pressure deficit is 0, so there will be no entry of water in fully turgid cell. इसको याद रखें कि fully turgid cell में वाटर की एंट्री नहीं होगी क्योंकि ऑस्मोटिक प्रेशर टर्गर प्रेशर के बराबर हो जाएगा, क्योंकि जो ऑस्मोटिक प्रेशर से पहले वाटर अंदर आता रहेगा. सपोज देयर इज ए वाटर एंट्री, सपोज इट इज अ नॉर्मल सेल and there through osmotic pressure water is enter into the cell, and as the water content increases in the cell, turgor pressure increases. शुरू में क्या होगा, ऑस्मोटिक प्रेशर से पानी सेल के अंदर आएगा, वाटर कंटेंट सेल का बढ़ता जाएगा, उस सेल वाल पे प्रेशर लगाती जाएगी, सेल प्रेशर बढ़ता जाएगा, और जैसे जैसे प्रेशर बढ़ता जाएगा ऑस्मोटिक प्रेशर कम होता जाएगा, और जब ये टर्गर प्रेशर ऑस्मोटिक प्रेशर बराबर हो जाएगा तो वाटर की एंट्री नहीं होगी. उस समय क्या होती है, सेल फुली टर्जिड होती है. तो इन फुली टर्जिड सेल ऑस्मोटिक प्रेशर इज इक्वल टू osmotic pressure and there is no further entry of water, so dPd is 0. Another case may be that the cell is flaccid. Flaccid cell is shrinked. Suppose it is a cell, it is shrinked cell, flaccid cell, and turgor pressure is 0 because there is no water. So Tp is equal to 0. So water move inside the cell due to osmotic pressure. So osmotic pressure is there, and then cell start to swell. अगर सेल फ्लैसिड है, सिकुड़ी हुई है, बिल्कुल पानी नहीं है, तो उसमें टर्गर प्रेशर होगा ही नहीं, बिकॉज देयर इज नो वाटर, सो देयर इज नो प्रेशर बाय वाटर. जब वाटर नहीं तो वाटर प्रेशर कहाँ से लगाएगा, तो दैट्स वाई टर्गर प्रेशर इज 0 इन flaccid cell. And when it is placed in a pure water, this cell, then water move inside the cell, and that there is osmotic pressure. So osmotic pressure से क्या होगा, सेल के अंदर वाटर आना स्टार्ट हो जाएगा. तो ऐसे केस में केवल ऑस्मोटिक प्रेशर होगा. सो नाउ dPd is equal to OP, osmotic pressure, minus turgor pressure, and dPd is equal to OP minus this turgor pressure is 0. So diffusion pressure deficit is equal to OP. So in flaccid cell, osmotic pressure is equal to diffusion pressure deficit. तो जितना osmotic pressure होगा उतना ही diffusion pressure deficit होगा flaccid cell में. और turgid cell में कोई diffusion pressure deficit नहीं होगा. And this dPd is responsible for entry of water. So this dPd we can say decide the dPd decides water intake of a cell. तो ये dPd से पता चलता है, अगर dPd ज़्यादा है तो वाटर इनटेक ज़्यादा होगा, अगर dPd कम है तो नहीं होगा. और मैक्सिमम dPd की होगी इक्वल टू ऑस्मोटिक प्रेशर. तो अगर और dPd डिसाइड होती है ऑस्मोटिक प्रेशर और टर्गर प्रेशर के डिफरेंस से. सो दिस इज द dPd, एंड द water potential of a cell is equal to negative value of this dPd, or we can say water potential is equal to minus osmotic potential minus turgor potential, or minus osmotic potential plus turgor potential. And in general, turgor pressure is positive value and this osmotic potential is negative value. So turgor pressure is in general positive because it exerts the pressure on the cell wall, so its value is considered positive. And osmotic pressure, work is done, so that's why, because water is entering into the cell, so work is done by the water, so that's why it is negative. And work is obtained in case of turgor pressure, so that's why its value is positive. So in general turgor pressure is positive, but in xylem cell during the transpiration it may be negative, but in general in cell it is positive value. And water potential of a cell is the negative value of dPd. That's why dPd is very important, and dPd is decided by osmotic pressure and turgor pressure. That's why both are important. Turgor pressure is decided by the water content of cell, is what high water content, more turgor pressure, and osmotic potential is decided by the solute concentration. If solute concentration is more, then there will be more osmotic pressure. So in a cell, water potential is decided by two factors mainly. What is the solute content? Number one, what is the water content? Then this difference will decide how much water can a cell take. तो किसी सेल के अंदर कितना पानी आ सकता है, कितना एब्जॉर्ब कर सकती है, ये डिपेंड करेगा उसके अंदर सॉल्यूट्स कितने हैं कि जो अफेक्ट करेगा ऑस्मोटिक प्रेशर को। दूसरा, उसमें ऑलरेडी कितना पानी है जो डिसाइड करेगा टर्गर प्रेशर को। अगर टर्गर प्रेशर बहुत कम है, पानी ज़्यादा आएगा। अगर ऑस्मोटिक प्रेशर बहुत ज़्यादा है, पानी बहुत ज़्यादा आएगा। तो दैट्स वाई दिस डीपीडी इज़ वेरी इंपॉर्टेंट इन कैलकुलेशन ऑफ और इन डिटरमाइनिंग द वाटर पोटेंशियल ऑफ ए सेल। थैंक यू वेरी मच।